Hello everyone, welcome to the channel Microbial World. So in the previous session, we discussed the bacterial cell structure. We studied the external structures of a bacterial cell such as the capsule, cell wall, flagellum, pili, sheath and prosthesis. So in this session, we will be discussing the internal structures of a bacterial cell. So first, let's see what is the cell membrane, which is also called the plasma membrane. Now, the cell membrane is the innermost phospholipid bilayer. So as you can see, if this is a cell, now this phospholipid bilayer, it is present just beneath the cell wall enclosing the cytoplasm. Now, if this is the cell wall, then this is the cell membrane. It is the innermost phospholipid bilayer. So it is present beneath the cell wall and it is enclosing the cytoplasm. Now this cell membrane, it is a very thin layer. It is 5 to 10 nanometer semi-permeable layer. Now what does semi-permeable means? Semi-permeable means it will not allow all the substances to pass through it. It will allow only certain substances to pass through it. Now, it will allow the solvents to pass through, but it will not allow certain solutes to pass through that layer. Now, this plasma membrane, it lacks sterol. Sterol, that means it is a lipid class. Sterols are not present. They lack sterol, but sterol-like compounds called hapenoids are present in the plasma membrane. And also, high proportion of proteins are there in the plasma membrane. Fatty acids are present, that is saturated and unsaturated fatty acids are there. Saturated fatty acids are there, also unsaturated fatty acids are there. And in addition, methyl, hydroxyl and cyclic groups are also present. The plasma membrane also has porin proteins. Now what is the function of the porin proteins? We have told earlier. Now this porin proteins, they are helping in the passive transport of nutrients and ions through it, through the plasma membrane. They are helping in the transport of nutrients and ions through the plasma membrane. Cytoplasm. Now the cytoplasm, it is a colorless, colloidal and viscous fluid. Organic and inorganic solutes are suspended in the cytoplasm. And as we have told earlier, the cytoplasm, in the cytoplasm there will be no membrane bound organelles. That is endoplasmic reticulum or Golgi apparatus or lysosome, peroxisomes. So any such membrane bound organelles will not be present in the cytoplasm. But there will be ribosomes will be there, mesosomes, inclusion bodies and nucleic acid. So all these kind of ribosome, mesosome, inclusion bodies all will be floating in the cytoplasm. Now the cytoplasm, it will not show any kind of internal mobility. They differ from this eukaryotic cytoplasm in that they do not show internal mobility, no internal mobility, or we can say protoplasmic streaming or cytoplasmic streaming. Now what is this protoplasmic streaming or cytoplasmic streaming? It is the flow of the cytoplasm in a cell due to the forces from cytoskeleton. Okay, so that is what is protoplasmic streaming is. Now the cytoplasm, it will stain uniformly with basic dyes. Ribosomes. Now this bacterial ribosomes are 70 S type. S means Swedberg unit. This is the rate of sedimentation in a centrifuge. Whereas eukaryotic ribosomes are 80 S type. Now this prokaryotic ribosome is made up of two subunits. A larger subunit called the 50S subunit and a small subunit called the 30S subunit. The main function of this ribosomes is to synthesize bacterial proteins and enzymes. And also these ribosomes are target site for antibiotics such as erythromycin, macrolides and aminoglycosides. Mesosomes. So mesosomes are vesicular or branched structures. It is formed by the invagination of plasma membrane. So as you can see, when the plasma membrane invaginates inside, mesosomes are formed. They represent eukaryotic mitochondria in function. And also these mesosomes are the site of action of bacterial respiration enzymes. It is more prominent or it is more commonly found in gram-positive bacteria. Mesosomes can also coordinate nuclear and cytoplasmic division during binary fission. Inclusion bodies. Inclusion bodies are believed to be storage food. So there are mainly two types of inclusion bodies, organic inclusion bodies and inorganic inclusion bodies. Organic inclusion bodies contain glycogen or polyhydroxybutyrate granules. Inorganic inclusion bodies contain polyphosphate or sulfur granules. Bacterial nucleus. Now this bacterial nucleus is also called nucleoid. This nucleus is not enclosed in any nuclear membrane. It is not enclosed in nuclear membrane and they lack nucleolus and nucleoplasm. It is represented by a double-stranded DNA molecule either in a coiled form or in closed circular form. Now this bacterial DNA, it can be found in the nucleoid as chromosomal DNA or it can be found outside the nucleoid as plasmid. Now this plasmid, it is an extranuclear genetic element. Now this plasmid is an extranuclear genetic element which is capable of self-replication. So this is all about the internal structures of a bacterial cell. We have discussed the cell membrane, cytoplasm, ribosomes, mesosomes, inclusion bodies and bacterial nucleus. Hope that this session was clear. Thank you for watching.